Good morning. My name is Ariel, and I studied X-ray crystallography of phosphomanimutase 1 protein in Dr. Karen Allen's lab in BU's chemistry department last summer. And as indicated by my thesis title, I spent a lot of time with this great protein called phosphomanimutase, and I will be sharing my knowledge on its physiological structure and its significance with you today. So phosphomanimutase, known as PMM, is an enzyme that catalyzes the conversion between D-6-phosphate and D-1-phosphate. So when a protein enzyme performs the conversion, we can also call this protein a mutase. And now you might be wondering how important these technical terms can be and why should I care about PM1? Well, PM1 exists in all our bodies. So here's a close-up look of the ER of an eukaryote cell. So here's like a cell, and there's the ER of it. And this is just a process in your ER. So where the arrow points to is where PM1 does its job of conversion, and it contributes to GDP monosynthesis, which initiates the pathway for modifying sugars in this diagram. So PMM is in the initial step of this process that is taking place right now in every single cell in your bodies, and without PMM, your cell will not complete this long mechanism chain and produce the necessary results. So that specific pathway is called end-link glycosylation. Glycosylation is a process that attaches a glycan, a sugar molecule, to a nitrogen atom in cells throughout bodies. It modifies the cell's surface and protein properties and is very fundamental to many important biological processes such as immune defense, viral replication, parasitic infection, cell growth, and inflammation. Moreover, PMM is important to be studied because it's disease-related. So congenital disorder of glycosylation, CDG, is a rare inborn error of metabolism that leads to deficient glycosylation of a variety of tissue proteins and lipids. And it causes serious, sometimes fatal malfunction of nervous system, muscles, and intestines. And then the symptoms can be observed all over the body. Your heart, you will have problems with your heart, liver, eyes, and everything. There are over 1,000 CDG cases reported globally, and CDG is caused by a mutation of PMM2 gene, and there is still no cure to it yet. Now, if you think back to my thesis title, I said my best protein friend is PMM1, but here is PMM2. How are they related? PM1 and PM2 are isoforms. So it just means that there are two functionally similar proteins that have a similar but not identical amino acid sequence, and they are actually coded by different genes. So PM1 was first found out in 1994, but it only exists in brain and lung tissues. So it doesn't correlate with the observation of CDG symptoms all over the body. Thus, it was doubted that PM1 is the cause for CDG. And there has to be something else. So scientists kept doing research until they found PMM2 in 1997 on a different chromosome, and PMM2 is expressed ubiquitously all over the body. To further support the speculation that only PMM2 is relevant to CDG, research group conducted experiment on mice. So they found out that mice deficient of PM1 lived and did not have observable phenotype change, while mice deficient of PM2 had no viability. So while the research on mutation of PMM2 is still going on, the result also suggests that PMM1 has a critical function other than D-1 phosphate synthesis in the end-link glycosylation and is independent of CDG disease. So in 2008, researchers found that the primary function of PM1 seemed to be related to glycolysis. So glycolysis is just a metabolic pathway that provides energy for our body to function in our daily life. So as shortage of oxygen and glucose occurs, ATP is depleted, and it leads to increase in a metabolic product called IMP. So IMP actually binds with PM1, and it activates PM1 from a mutase, which is the function of PM2, to a phosphatase. A phosphatase just means it's a phosphate removing group. So if you look at here, there's glucose 1,6 biphosphate, so there are two phosphate group. And after PM1 binds with IMP and being activated to a phosphatase, it removes one of the phosphate group and turns it into glucose 6-phosphate, and this is part of the pyruvate metabolism. So it provides another pathway for pyruvate metabolism during ischemia, and it seems to have some medical application on that part. So the purpose of my senior thesis project is just to determine the physiological structure and functionality of PMM1, as well as the structural difference between PMM1 and PMM2 that allow only PMM1 to bind with IMP and switch the activity from a mutase to a phosphatase. And this is just a graph showing the PM1 reaction mechanism. We're trying to have a crystal structure of this PM1 binding with the IMP so we can see how they react with each other. So we decided to use X-ray crystallography as our method. X-ray crystallography is a technique used to determine the atomic and molecular structure of a crystal, in which the crystalline atoms cause a beam of incident X-rays to diffract. So you expose it to an intense beam of X-ray, and you can observe the patterns in a screen behind the crystal. And then by measuring the angles and intensity of these diffracted beams, you can acknowledge the position, chemical bonds, disorder, and various other information, and you would have an electron density map. And then by doing computer data analysis, you can have a 3D atomic model of your molecule. Since many materials can form crystals, such as salts, metals, semiconductors, as well as various inorganic, organic, and biological molecules, X-ray crystallography has been fundamental in the development of many scientific fields. It has contributed to revealing the structure and function of many biological molecules, including vitamins, drugs, proteins, and nucleic acids such as DNA and ribosome. So although X-ray crystallography sounds groundbreaking and the process seems straightforward, it is not so easy to accomplish. So in order to perform X-ray crystallography, first we need crystals. And growing crystals can be time-consuming, difficult, and sometimes your protein just hates you and will not cooperate even though you're using the same condition, but the lab environment is not constantly stable. So you would need a pure protein droplet, and by vapor diffusion with the well solution, the liquid would precipitate to solid. So there are two methods: is a hanging drop and sitting drop is kind of straightforwardly demonstrated by the graph. It just one of them, the droplet is hanging and then precipitating with well solution, and the other one just sitting there in the well. So it is extremely difficult to predict good conditions for growth of well-ordered crystals. This is why we use a technique called screening. So we would prepare a very large batch of proteins, usually kind of a 96 wells, and then a wide variety of crystallization solutions are tested. And because screening is the best option for acquiring PM1 crystals and it is in batches, we need to first obtain a lot of pure PM1 protein liquid. So here's my procedure and workflow. So we would first genetically engineer E. coli bacteria. So we would first ligate PM1 gene with a vector, and then we would insert the vector into E. coli bacteria and transform the E. coli bacteria so it has the genetic coding to start making PM1 proteins. And then we would put E. coli, the transformed E. coli, into a culture and grow, and we would induce it with IPTG. It's basically just a command telling E. coli to make lots of proteins, only proteins, no other things. So then after E. coli finished growing, we would take them and lyse them with microfluidizer and ultracentrifuge. So basically they just break the cells and spin down all the cell content, so only we are left with the liquid, but we would get rid of the cell walls, debris, and other contents. So once we only have the protein parts of the cell, we would try to separate PM1 protein from all the other proteins in E. coli. So we run the protein liquid through fast protein lipid chromatography machine, FPLC machine. So it's just like a really strong filter, and we have 3 columns: His column, ionic exchange, and size exclusion column. So we would have the column attached to the FPLC machine, and these columns target specific traits of PM1 protein. So once the protein liquid runs through, the column would grab onto the PM1 protein, and then we would wash with elution buffer, so the protein would come out of the column, and then we would collect those. And after 3 steps of purification, we would have generally a very pretty pure protein liquid concentration. And then, as I mentioned before, we would first do a screening batch. So it's 96 wells, it's a lot of different conditions. And once we see a good condition that shows signs of crystallization, we would take it, optimize it in a 4x6 grid screen, and then we would keep optimizing the conditions until we have a diffraction quality crystal. And then it just basically doing crystallography to the protein until we have a structure. So here are our actual crystal setup tables. So this is so the shaded areas were the conditions where we took from the original screening batch, and we optimized it in varying concentration of salt versus varying concentration of proteins. And the stars just shows that there were crystals. And we took the exact same conditions, but we did optimized it with varying concentration of salt versus change in pH. And again, the stars were showing where the crystals were. So here are the actual pictures of our PM1 protein crystals. I think they're beautiful and amazing. They're all needle looking like. So it's kind of cool. Like, you don't expect, I thought like originally it's gonna be like, I don't know, like the crystals you see like in cool pictures, but you know, they're still beautiful. So we looped one of the crystals from the first table, well B3, and subjected it under X-ray at 0 degrees and 90 degrees. So we just have like a really nice, like around 3D picture of it. And the resolution quality is 3.5 angstrom. So the resolution quality is more than the less than 2 angstrom standard for an accurate computational modeling. So the smaller the number is, the better the quality of the diffraction pattern. It just means that the protein folds are likely to be correct in general, but some surface loops might be mismodeled in the product. However, already from this diffraction pattern, we can tell that the structure is similar to the published results of PM1 structure. So this published structure of PM1 concludes that PM1 protein structure combined with kinetic experiments suggests that IMP binds PM1 with high affinity and converts the enzyme activity from that of a mutase to a phosphatase. And it might just be due to differential conservation of residues between PM1 and PM2 in the IMP binding loop region. So it does support our hypothesis of the primary function of PM1 protein. Unfortunately, PM1 research has been a fairly new study subject, and this is what we have on it so far. We don't know exactly why PM1 reacts differently, because the published crystal structure as well as our crystal structure are open conformation. So open conformation just means PM1 and IMP did not have substrate binding in the crystal structure. They only co-crystallized at the same time, but separately. So one of the things for future research is that we would try to explore and optimize more crystallization conditions for close conformation so that we can see the structural relationship and mechanism of PM1 and IMP interacting with one another when they do bind. And another improvement is just to use a better X-ray source. So once if we have a really good diffraction quality crystal, we can send it to a professional X-ray source. So it's kind of just taking a picture of the same object with a better camera so we can see it better on the computers. And lastly, because I don't really have any significant results or conclusions, I would just like to end my presentation by sharing my reflection on my experience working in the lab. I think X-ray crystallography is just fascinating in that it unveils the physical and chemical information of protein molecules that we cannot imagine with our eyes. The process is kind of elaborate and complicated, but really it boils down to a trial and error method. You try something today, if it works, great. If it doesn't, move on to the next condition. The field of X-ray crystallography is about the quantity of experiments performed. So I was really honored to be offered to have the opportunity to work all summer, Monday to Friday, 10 to 5, but it was pretty much just trying and failing every day until the last week of August. So the preparation of the pure proteins actually takes 2 weeks, so that was this whole workflow until we get to pure protein. All the steps would take 2 weeks, and then after setting up the crystal trays, the crystals would take 2 weeks or so to grow as well. Just imagine the frustration that after a full cycle of workflow, that nothing really shows up, your protein just keeps dying, that you can't even set up a crystal tray. And I think that the practice of failing and having to restart again and still be positive is not really incorporated into high school labs, not even bio labs in BU, or addressed in senior thesis usually. But failing is the real lab science for 99% of the time. I just want to bring up the backstories to highlight the fact that you can't expect that during research immediately finds a conclusion or a impressive result. Every small scientific progress is achieved through long-term dedication and efforts. And this is why we should hold so much admiration for scientists out there who repeatedly fail but kept going in lab every day. So here's my bibliography. I would like to thank Dr. White for coaching my thesis writing and presentation practices throughout the school year. And Catherine, who's a chemistry PhD candidate, she's my lab mentor for being such a great and patient mentor for me and struggling with me about dying PM on proteins. And Dr. Allen for offering this opportunity for me to grow cool crystals in her lab. And I'll be happy to take any questions.
Fabulous end to all of our senior thesis presentations this year. Lovely, Ariel. Questions for Ariel? Oh, sorry, yes.
In the picture you showed the protein, there were no side chains. Position side chains, getting connection.
Do you mean this one?
Yeah.
Oh, I'm just, what was your question again?
Get information to position side chains.
I think the side chains were correct, but what we were looking at is just cap domain. So this is where it would bind with the IMP substrate, but we're still looking for conditions where they would bind. So in our crystal structure, it's more like PM1 co-crystallized with IMP separately, so we can't see them binding together at this site. But this information is correct, and we know about it.
Other questions for Ariel? I have one. So this was a chemistry lab, but how much, what was the breakdown, do you think, between you doing biology and doing chemistry in this lab?
I think the concept is biology, and it's more biology application. But I think the lab process is chemistry, preparing the lab and doing it. But I think there's biology part to it, the transformation of E. coli. Pretty much everyone did that in Bio 108 labs, I think. So it's a good mix of biochemistry, I guess.
Any final questions for Ariel? All right, I think Dr. Betchoff will wrap us up.